Hi all, hope you are all fine and safe. My name is Sridevi Suresh Kumar. I'm heading GATC Lab, and this is my web address. If you guys want to know more about my projects, please visit my website. My lab is primarily working on triplet repeat expansion in model plant, Arabidopsis thaliana. Before explaining my research, let us brush up relevant fundamental details. What is triplet repeat expansion? So changes in the copy number variation, especially in trinucleotides known as triplet repeat expansion, affects gene expression and as well as the protein function. So far we know more than 40 plus neuronal genetic disorders in the humans are attributed to triplet repeat expansions. So the experience from the plant side, from looking at the phenotypes and molecular understanding, it's more and more clear that the neuronal cells are vulnerable to the repeat expansions in the humans. So it means that the problem lies in understanding the fundamental mechanism that could be addressed using plant model. So that's where I put up this question, why do we study in plants? But there is also another problem why so far we haven't understood the mechanisms, because lack of animal model to study human triplet repeat expansions. So I see it's a long-lasting struggle in the field. So now we have Arabidopsis thaliana, FSL model to understand gene silencing caused by intronic repeat expansions. So in this next slide, so what we see here is, this is the basic structure of the gene: promoter, 5' UTR, followed by exon, intron, and 3' UTR. So what we see here, so these are all different permuted triplet nucleotides. Expansions in this nucleotide causing these neuronal disorders. So this triplet repeats expansions could occur any part of the gene. Can be 5' promoter, exon, intron, or 3' UTRs. And there is a differences between the repeat expansions in the exon compared to the other part of the genes. So particularly I'm taking intron as an example here. So exonic repeats tend to be shorter, and it is easy to engineer, easy to clone and engineer and develop a model. So coming back to this, either intronic repeats or the repeats occur in 3' or 5' UTR, they tend to grow much longer, and the repeat length is very, very long. So you cannot clone them because the bacterial system or the yeast system won't allow you to clone these longer repeats, and also involved in the gene downregulation. So in case of exonic repeats, so what happens here is, so you might explore Huntington disease. So Huntington disease is due to this CAGC repeat. So what happens is, if you have this particular repeat codes to the particular protein and increase the number of the protein, which increase the number of the amino acids, which leads to a toxicity. But if you see in the intron or 5' or 3' UTRs expansions, they tend to downregulate the gene expression. So here, particularly, I would like to talk about the intron repeat expansion called Friedreich ataxia and also called FRDA. This is quite similar to what we have discovered in Arabidopsis thaliana. So this striking parallel between Friedreich ataxia and the phenotype which we found in Arabidopsis. So if you take human case, in the humans and Arabidopsis, the common between both is this GAA-TTC repeat expansion, and this repeat expansion occurred in the intron. So if you take in the human's case, it is in the first intron of frataxin gene, and then downregulation of the frataxin gene because of the repeats, longer repeats, and led to a Friedreich ataxia. If you take an Arabidopsis gene, it happened in the IAL1 locus. This is a gene, and then downregulation, the expression is low, and then lead to the L phenotype. So in a moment, I will show you how the yield phenotype looks. So this is how wild type Arabidopsis thaliana looks. And this is how the yield phenotype looks because of the gene downregulation. So this discovery of the phenotype is published quite a while ago. So if you guys want to know more about this phenotype and the gene, please look into this article and you can able to find the relevant information. So what's next? So we have a very strong question to follow up on. So how does this triplet repeat expansion leads to the gene downregulation? So one of our PhD student and inputs from honors and masters and postdocs, what they found is that there are small RNAs are formed from the repeat expansions. So and this small RNA, you might know about this different types of small RNA. It is also present in humans, and it is also there in fly and other model organisms and plants as well. In fact, this small RNA is first discovered in the C. elegans, and you guys know about the story. So what we have done is that what we found is the small RNA mediated pathways involved in the gene downregulation. So these are the components which are involved in this pathway. So we have knocked down all these components using RNAi technology. You can also use the CRISPR technology as well. So what we found is that when we knock down each and every components in the yield phenotype, what we found is that the phenotype is suppressed and then the gene expression is increased. So that makes us to understand that this is the mechanism indeed involved in the gene downregulation. It is mediated by the triplet repeat expansions. And so we have still several fundamental questions remain unanswered. So I have formulated a couple of questions. We have many more questions to be addressed. Join us to explore. Thank you.